clocks around Europe are coming to the hour, so it's time for us to open the Euro-BioImaging pub session. So welcome to the Euro-BioImaging pub, which is a very informal gathering. For those of you who have not been present, this is we throw away all the titles and step down from our high horses and then just enjoy enlightened presentations. So this is a pub where the drinks and refreshments are on the house. The issue is that it's on your house, but the entertainment and the enlightenment is provided by us. And if you haven't been here before, if you want to receive information about the pub, please put your name in the chat box, and then you will be on our mailing list. And just for your information, this session is being recorded. We have a few announcements, first of all, but I think I'll go ahead and present our speaker for today, and then we can go to the announcements. So today we have Peter Horvath, who I know very well because he has been working in Finland at the Finnish Institute for Molecular Medicine in Helsinki, but he is also working at the Institute of Biochemistry at the Biological Research Center of the Hungarian Academy of Sciences in Szeged. So he's been holding a Business Finland Finnish Distinguished Professorship title at the FIM Institute until 2018. He has a very broad background in terms of what he will be presenting today. Life beyond the pixels, because he has been working as a software engineer and as a mathematician, receiving his PhD at the University of Nice, Sophia Antipois in France, working on satellite image analysis, and he has also worked as a senior scientist at ETH in Zurich and at the Light Microscopy Center. So he has a very heavy engagement and interest in computational cell biology problems, 2D and 3D image analysis, development of different microscopic image correction techniques and so forth, and now also machine learning and machine learning in the context of high throughput. So that was my introduction of Peter, but before...
Thank you very much for your kind introduction and Euro-BioImaging. Thank you very much for inviting me for this talk. And I have quite a long history with Euro-BioImaging. I was there when the Swiss bioimaging started, probably 12-ish years ago, and I'm German, and now I'm one of the head of the facilities in Finland, and also actively participating in the Hungarian one. So I really appreciate that this RIA exists, and said thank you very much for inviting me. So I'm going to talk about pixels and how we interpret them. But before I go, let me just show you a video. So if this video was taken probably 12 years ago, and this is an siRNA screen of bacteria, where we were interested in whether if we silence genes in cells, what's the effect of bacterial infection. And if you think about what the amount of data, what comes out from this screen, so that was a genome-wide one with like 20,000-ish genes, all in 4 repeats and 10 images in different channels. It's millions of images, billions of pixels. And if you want to successfully manage such a data, then you should design precise, fast, and intelligent methods, because it's just simply impossible to like manually handle. And I should also say that in the last 12 years, all the technologies advanced, all the throughput advanced, so we tackle with much larger and more data. And therefore, I actually established my research groups called the BioMag groups, Biological Image Analysis and Machine Learning groups. And we work on 4 major areas. The one is image quality improvement, photo bleaching correction, flat field correction. We deal with image segmentation and tracking. Probably this is what is closest to my heart. Then we developed several machine learning approaches to phenotypically profile single cells, and lastly, we work with single cell isolation. I'm not so sure if my Zoom window is on the front of my screen or not. Hopefully you don't see it. I do. So the large topic we cover is isolating single cells using correlative microscopy. And I'm going to just guide you through these 4 fields and show what we developed most recently in these fields. So to start, if you see a picture like this, you probably see columns, couples, a beautiful sea. Probably your human eye does not recognize that the corners of the image is much darker than the middle. If we would make an intensity measurement here, it turns out that the corner is 3 times darker than the middle. This is probably not surprisingly also there in microscopy. And I just collected here a couple of examples, like mouse kidney stitched, yeast cells, cancer cells, and you see that if we try to quantitatively analyze these things, then illumination problems have very serious effect, actually even falsify image analysis. So we need to handle that. And as we realized, I was working in a big microscopy facility before, and we realized that most of our users are not aware of this, and even if they are aware, they don't really handle this problem. So we started to design a method called the CIDR, that is a general image illumination correction method, where the idea is, I'm not going too much into the mathematics of it, but the idea of CIDR is that we have the reality. The reality goes through an optical, or let's put it like that, an observatory system, which is usually optics and digitalization way, like a CCD camera or so, or a CMOS chip, and then the original image becomes a distorted image. And the question is, can we somehow turn back this image to the original performance? And what we said is, if we would have infinite amount of those images, we would put them together, and we would observe at any given pixel position the histogram, then we should observe exactly the same distribution of the pixels, basically the same histogram. If it's distorted, then these histograms are falsified, and then we developed a method that actually brings them back to the same. And of course, we don't have infinite amount of images, so we had to tackle with this like leaky data. We used regularization techniques. So then CIDR was born, the method we proposed. We tested on different microscopy techniques, and it largely outperforms any state-of-the-art correction, and also highly dominates over calibration techniques. So we tested on epifluorescent, bright field, DIC, and super resolution, and also on like real life images from the National Gallery, and it works pretty stable. So I recommend you to try either the Java, MATLAB, subprofiler, or Python implementations we have. So once the images are corrected, then the next step, what we do is process them. So basically to get some sort of quantitative information out of those images, And for that, I'm going to now use this picture we took several years ago about influenza infection and different drugs affecting influenza. So usually we deal with fluorescence images where fluorescent particles are put in different cellular compartments, and usual steps we do is detecting cells, detecting their compartments, and extracting their features to get quantitative information. So basically, turning an image into a segmented image is the first step, where we identify the nucleus, we identify virus particles one by one, and probably also cytoplasm or some other cellular compartments. Once we have these segments, then we measure their morphologies, how big they are, what's their area, their intensities, intensity distributions, textures, what type of like patterns they have on the images. And given that, basically we turn an image into a set of numbers. Usually it's a matrix where every row is one cell and every column is one property. But so the thing is so easy as it gets, or well, actually apparently not. So there are very interesting microscopy techniques where single cell segmentation becomes really challenging, like DIC or label-free microscopy, phase field, and there are other label-free microscopy techniques, or when cells are sitting on top of each other, when cells are touching each other. And these are problems what is very difficult to solve, and this is where my group did probably the major contributions to the field. So I cherry-picked a few methods we developed during a couple of years. So the first one is when cells are sitting on the top of each other. So many of you are physicists or microscopists, so you very well know if the point spread functions, the resolution is bad enough, and if we put two cells on the top of each other, then because it's an integrative technique, the intensities of like double density fluorophores makes the intensity is measured double. So if there are two cells on the top of each other, then the junction is twice as bright as normally, as you can see it here and there. So we said that we are looking for shapes that have a shape of a cell. So it's usually like elliptical or circular. And if they are on top of each other, then we allow them to have double intensities than normally. So we created this model, and with that we were like very successfully able to isolate or like identify the borders of the single cells. This has been used, for example, in Finland for like detecting single cells on breast cancer TMA spots. We used it for several other single cell phenotyping tasks. The other thing, what very often happens in microscopy cellular images is that cells are not sitting on the top of each other, but very heavily touching each other. And if that happens, we actually made a mathematical model and observed that in these touching, there are two very exciting points. These are these corner points where the contour actually suddenly breaks and changes direction. So its curvature, it's basically, it's like smoothness changes. And if we said that if there are these high curvature places opposite to each other in a good proximity, then why don't we just like create a cut? And for that, we use the so-called active contour models. So active contour models are like, it's basically, you can imagine as like a gummy stream or so that has energies, and it want to minimize its energy, and we can define what is an energy. Like, for example, it should sit on the edges of the cell. Or we created energies so that if we have this configuration that two curvatures opposite to each other arise, then they force to like cut. And this is what you're gonna see here. This large curvature place is opposite, and then it cutted them, and then we got the single cell resolution. And this is what we did in the last 10 years. All of a sudden, this magic buzz, the deep learning word came, and it changed. I think I can say it changed everything. So for me, the big start was the Kaggle Data Science Ball competition in 2018, where the task was to design method that actually finds nuclei on diverse images, and you see the diversity of the images. So it was a worldwide competition, and you were actually required to create one particular method that actually works for all of these type of images and finds nuclei the best performance it can. And it turned out that this is an interesting problem, and it became probably the biggest ever bioinformatics challenge worldwide, with nearly 4,000 participating groups. And we said that, okay, this competition is for us. This is what we do since 10 years, so we should win it. And sadly, actually, our methods didn't work very well on these very diverse images. So we were at position 1000-ish or so. And of course, it turned out that everyone uses deep learning in the first 1,000. So we also started to use deep learning, and with that, we get probably in position 50 or so. So it's like not that bad, like reasonable position. So it turns out, probably you guys are familiar with deep learning. This is a very fantastic umbrella of technologies, but there is one very big requirement, is that you need training data. And if you don't have enough training data, then the performance is, let's say, mediocre. And we didn't have too much. We just had as much as the others. And this was a couple of hundred images and a couple of thousands of cells. And large players like Facebook or Tesla, they play with millions of images. So we said that, okay, can we somehow boost our training set? And we started to use a technique called image style transfer learning. Image style transfer learning is a technology where you have a basis and a style, and you can copy that basis as it was painted in a given style. Or probably you saw these pictures, a German city, a famous painter's paintings, and then as the city was painted by the painter. Okay, so can we somehow use this technology to create an endless source of training data for ourselves? And we said, yeah, let's try. So we said that let's pick a real microscopy image, and let's ask the program to put a cell here and here and here, or there and there, or go on. And from real images, we created fake images where we could control the program where the cells to be. And with that, we started to create millions of fake images and started to train our methods with that. And then the nucleizer method was born, which actually got the best position on best score, and still holds probably the best score on this competition. And I believe this is probably to my best knowledge, this is the state of the art in single cell segmentation. You see a couple of examples here that it is can be used very widely, but we have models for all very classical and exotic microscopy techniques already. So if you go to nucleizer.org, then you can try out, you can just drag and drop your images and then can try on the fly how the method works, and you find a couple of pre-trained methods as well. Yeah, so deep learning was fun. So currently we are working on extending our deep learning capacities in several fields. So one very exciting things was in this year that we integrated nucleizer into the NAPARI, that is driven by the Chan Zuckerberg initiative. So we were participating amongst another couple of methods in integrating existing nuclei detections into NAPARI. It's done, and one can already download it and try out, not only predicting but training. We work a lot on three-dimensional annotations, like one-click annotations, and also new representation techniques, like Fourier-based representation techniques and other spline and active contour representations to have a more flexible and fast single cell detection. And with that, I'm nearly finished with my deep learning part, except that I wanted to show you one very exciting project that as such, it's not really exciting. And especially if you ask the PhD students who are annotating astrocytes on tissue sections, they probably say it's not exciting. So what happened is that one of our Finnish collaborators, group Mart Sarma's group, asked us if we could make like a tool that actually counts astrocytes on sections. And then, yes, we thought that we could develop something. This was back in the days when deep learning was probably not there or not so premature. So we started with classical techniques and achieved, I would say, like mediocre results. But then two of my PhD students started to work on the so-called detector networks that actually are used to like find instances, not to segment, but find instances on pictures. And they started to train a deep learning method to find these astrocytes. So you see, it's not very easy. And it turned out that they spent like days and trained 6,000 single cells, and there was not really good results. And when we reached, I think, 12,000 or so, all of a sudden it started to work, and it started to work pretty well. And what happened is that we asked other PhD students from the same lab if they could annotate the same images and highlight where the cells are. This is the result. A, B, and C are 3 PhD students. They actually got about 80%, 75% accuracy in like reproducing each other's results. But then we asked this PhD student A to make this 12,000 single cells, and then we trained the machine, who is D. And it actually turns out that the machine can learn PhD student A better than its classmates. So I'm not saying that deep learning is changing the world. Well, probably I said that already, but it is very, very good for certain tasks that are often very complex tasks, and I really recommend to try it out. But okay, let's get back to our mainstream. So we said the influenza images, and we got numbers out of them. All these 15 minutes, what I spent in the last 15 minutes was like showing you what kind of additional methods exist to find the single cells. But basically, almost always the problem or the task is that you have an image and you turn it to numbers. But this is not really what my pathologist, oncologist, and biologist collaborators really want. They don't care big matrices. They need to know whether a drug will like cure a patient, whether a drug or an siRNA or a depletion has an effect in biology. So turning these big matrices to smaller matrices, we have a project called the Advanced Cell Classifier. The Advanced Cell Classifier is a program to provide very accurate analysis with minimal user interaction using advanced machine learning methods. So it's basically a phenotyping tool that utilizes machine learning to get phenotypes from microscopy images. So Advanced Cell Classifier works like this. You have a picture. In this picture, what you see is primary renal tumor, like patient-derived cells, and they have one marker. It's a Ki67 proliferation marker, and they are drug-treated. And what we are excited about is that these primary cells can be cured or stopped by oncology drugs. So what, for example, here we were interested at is what is the proliferation rate after exposing the cells to drugs. So what we do with the program, we say that, hey, this is a proliferating cell. You see the Ki67 green marker. This is a non-dividing cell. This is something else. And by training this, the machine can learn and can predict to unseen cells. And what you see here is that machine predicted that number ones are the non-proliferating number twos, and the proliferating number threes are some feeder cells. And basically, we can apply this training of the pathologist to a large set of even billions of cells, and then can give like an efficacy of a drug or so. So yeah, this method was published, well, amongst other publications in 2017, and then most recently, about 2-3 weeks ago, we published its further developed version, the regression plane concept, that analyzes phenotypes in a continuous manner instead of like discrete classes. So we said that biology is not discrete. You very rarely have like settled states of cells, but cells are rather move on a continuum. And then we created a new interface that is called the regression plane, where you can drag and drop your cells, and instead of classes, the machine can learn positions on a continuous transition. For example, a very exciting example is cell mitosis. I'm sure many of you are aware of the MitoCheck project, where Ellenberg et al. And other colleagues were checking the cell mitosis and defined, I think, 7 or 8 states of mitosis. We said that, hey, let's consider mitosis as a continuum, and instead of putting them in discrete classes, we can consider as a continuum, and the regression plane concept can analyze cells as they are moving, and then can highlight like much more refined states than just these 8 classes, or more recently like 15 classes of mitosis. And at the moment, what we are doing, it's an infinite resolution gain of cell division. Right now, what we are doing is we are establishing the proteomic map of mitosis at an infinite resolution using this method. I'll tell you a little bit more later. And some time ago, with Ann Carpenter from the Broad Institute, we decided to put all these developments, what I've been showing you, like the identification, segmentation, classification of cells, to a web, and then we decided to implement the PIXIMI, that is a web-based single cell single cell phenotyping tool. You can find more details in cyto.ai, but basically it utilizes the GPU capacities of your web browsers through your web browser, your computer, and then you can do single cell analysis on the fly. And now I'm going to show you a few examples where we use, or where we have been using the current developments. So the first I want to show you is a project together with Jean-Pierre Bouquin from the Children's Hospital in Zurich, where we are interested to find drug treatment strategies for leukemic children who suffer from ALL, and using high content screening. The idea what we had is that let's take bone marrow cells, let's put drugs on them, and then let's put the leukemic cells on the top of them, and then let's see what happens to the leukemic cells given that we treat with some drug or we deplete some genes. And the problem is that this is a living system, and it was very difficult to find fluorophores that actually act and could paint the different cell types differently. So we basically had to find the fibroblasts and the leukemic cells on the same image, but given they are painted with one color. And then we used deep learning for that. So you see these are like fairly complex images, and then we can train machines that actually can distinguish from like between leukemia and the bone marrow cells. And then of course the hematologists are interested in what is the remaining leukemia versus MSC cells after drug treatment. So we established a company as well as spin-off company, the Single Cell Technologies, that actually provides professional solutions for that, and for example for this project as well. So we have like a professional solution as well that actually can handle these with a one-click solution. So like this is the singlecelltechnologies.com. It's an image analysis company. Another project I wanted to show you is what we do in my Finnish group is image-based personalized medicine and translational medicine for solid cancers. So the original idea we had is that we have the patient, we take oncology sample from the patient, create primary cell lines, and then use drug treatment to analyze basically, so to treat cells with drugs and profile what's the effect of these oncology drugs. So we have 550 oncology drugs available at the Finnish in the Finnish Institute, out of which 150 are oncology drugs approved by FDA and another 400 after or second phase clinical drugs. So we treat the cells in different concentrations, do the imaging, bioinformatics, and report back to the doctors. So whereas our leukemia pipeline was used to treat several hundred patients, here I cannot report such a great success yet, as it's really difficult to create cell models from an oncology sample. So we actually had probably a dozen patients where we applied this technology on. And more recently, we actually realized that two-dimensional biology is probably not appropriate for all type of like human disease screening. So I just decided that we will create a full-featured 3D single-cell screening pipeline that generates organoids, selects those that are particularly suited for experiments, do imaging and single-cell image analysis. And I would like to show you our current advancements we have in 3D high-content screening. And I should actually tell you that basically we are ready with the first platform. So one of the first steps we developed using a so-called attract grant. If you don't know, I really recommend you check it out. This is a technology development grant of the European Union. I believe that this will also be part of the New Horizon program. So they helped us to develop a prototype and bring it to like market ready. And the idea was that if we can generate spheroids, we would like to select those spheroids or organoids that are particularly suited for like drug testing. So basically they have like uniform circularity, size, roundness, and so on and so forth. So we created a small device that actually images them, deep learning analyzes their like look, and then a small manipulation robot goes there, picks up the single cell and puts it to a screening environment. And let me just show you the video how the machine works. So this is the setup. It's a stereo microscope, very cheap one. A lot of different XYZ motors and some electronics and computation. The microscope takes pictures, deep learning finds cells. Then based on some criteria, we decide whether a certain spheroid or organoid is suited for further analysis. Like here, the criteria was like an area criteria, and then like 2 of them are appropriate. And then comes the small needle, picks it up, sucks in, and then puts it to the target space where we can do the drug treatment, and then later on prepare it for imaging. So yeah, this is like completely a custom development, even the holders, the robots, the software, everything. And then it can just really make a very uniform plate with like same size spheroids. The next step, what we are interested in, is imaging. Imaging large things in high throughput is very difficult. So confocal microscopes are very limited in 3D. Light sheets are also very limited because of like known problems that you need light penetration from 2 sides and very so we actually decided to go with Leica. who has this new idea of light sheet probably know that there is a mirror attached to the objective. So basically the light sheet side and the observation side is just not 90 degrees, but 180 degrees. So you can generate certain cell types that actually are sufficient for high content screening. And this is what we are working on, and it's basically ready. And with that technology, we can go up to like 500 microns at a single cell resolution in certain cell culture organoids or spheroids. Of course, the biggest problem is analyzing the data, and this is where we work the most. So we decided to go like purely deep learning, but for deep learning we need annotated data. So first of all, we had to create an annotation tool. We created the 3D cell annotator software. You can get it 3dcellannotator.org. That actually, like with 3-4 clicks, you can annotate a single cell. And with that, we were collecting a bunch of spheroids, and we were just like recently, a couple of weeks back ago, like ready with our deep learning segmentation tool that can really get the single cells from like larger spheroids, and then we are right now already able to phenotype them at a single cell level. So the system is up and running just ahead of publication. And for example, we use this technology in another Horizon project together with the Heidelberg clinics to measure drug efficiency in pediatric brain tumors, where we are interested in what are the drugs that actually can catch the brain tumor. And now the last part of my talk is about single cells, how to isolate single cells from complex or less complex environments. So I was talking to you about single cell detection, segmentation, phenotyping, and we thought a few years ago that if we can find very efficiently a single cell in an environment, why cannot we just get it out for molecular analysis? And we developed the CAMI system. The CAMI system is computer-aided microscopy. It gets the microscopy pictures, segments them, finds the phenotypes, and then laser capture micro dissection machine is controlled micron precision there that cuts out the single cell and collects it for molecular analysis. So in the paper, we presented RNA and DNA sequencing at the single cell level. And to show you how it works in real life, and what we are currently working on is single cell proteomics together with Matthias Mann's group. You can find the or very recent publication it's still in bioRxiv, but Mundetol, and you see how this CAMI system isolates the single cells from tissues and from cell cultures for single cell proteomics. So they are basically collected in 384 well plates and then analyzed. We also work together with the Pathology Institute in the Zurich University and also with WHO to trying to redefine cancer grading. For example, what you see here is renal cancer, and what we are working on is to like fully automatically detect single cells in the human cancer tissue, cluster the cells, isolate, and not only morphologically but also like molecularly describe these cells. And with that, actually, this brings me to this deep visual proteomics that I was mentioning. We do with Matthias Mann and Emma Lundbeck together, where the idea is that we take human tissue, we process it with artificial intelligence, get the single cells, and do ultra-sensitive proteomics, and then clinical knowledge graph, and make sense of this proteome data as well for clinicians and biologist researchers. And my very last publication in single cell, it just appeared 1 or 2 weeks ago as well. It's the AutoPatch system, where the idea is or the question is, can we get a single cell also from a human brain, like a it's not a living human brain, but like a freshly operated human brain, so that we use patch clamping, fully machine learning guided. So a human brain under the microscope freshly looks like spaghetti. You have the cells barely seeable, but if you can detect them, then you can put a pipette and get them out or measure them. So we decided to control this pipette automatically to the cells and then suck them in and do electrophysiological measurements and also isolate them. So we did the whole stuff with deep learning, and what you see here, it was never touched by a human. A machine selected a cell deep in the human brain. A small pipette goes there and starts the electrophysiological measurements to phenotype the cell, and later isolate the cellular content that we do together with the LM Brain Institute in the US and sequence that. So you can imagine how many difficulties we have. Like if you hit another cell, you need to just make a shortcut. You need to control 6 motors at a time in a very plastic environment that is like not very easy. So we have like real-time trackers on the cells and so on. So that was more or less my talk, but I still have 2 quick topics. The one is that we were very actively there in the COVID fight with single cells. So with the Human Lung Atlas Consortium, that is part of the Human Cell Atlas, we participated in describing ACE2, describing where the entry factors are expressed, and probably what we are the most proud of, we were also a part of this study discovering neuropilin-1. This is what is another host factor for SARS-CoV-2 besides ACE2. And we did that with single cell microscopy and single cell data. Now, basically the same methods I'm showing you today. And if you are interested in the topic, then we published several review papers in the last couple of years about screening, data analysis strategies, phenotyping, 3D high content screening. So just feel free to either contact me or just try to get them. And they are quite helpful, I think. And I thought that I shortly present my unit at the Finnish Institute for Molecular Medicine, that is the so-called high content analysis FIM-HCA unit, where so this is a technology center. It's located in Helsinki. It is also part of the Helsinki bioimaging platform, part of the Euro-BioImaging. It's an open access imaging core and mostly high content screening and high content analysis, with a couple of faces you probably already know, Antti Hassinen, Vilja Pietiainen, Lassi Paavolainen, and Mintu Poso, who are my colleagues working on serving high content needs, mostly 2D, but more and more 3D, and also single cell isolation. So we have PerkinElmer, Opera, Phoenix. We have microdissection machines, smaller screening machines, and as for applications, we deal with solid tumors. We do this so-called drug sensitivity and resistance assays, this DSRT, to test drugs on patient-derived cells. And I think you probably know the Opera Phoenix. I don't need to show you. We have this fantastic machine with robotized, fully robotic arms and 24/7 working on 2 or 3D imaging. And we also work together with DHT, with the high throughput biomedicine unit, that is an automated liquid handling facility, to not only do the imaging, but also together with them, we do the assay handling and plating and so on. They are equipped with all sorts of very fancy robotics. And with that, I would like to thank my Finnish group, my Hungarian group, those who gave money, the collaborators, and I really appreciate your attention. Thank you very much.